Hey, thanks, and thanks to the organizers for putting this all on. It's really stimulating to see all the different ways that that these two disorders that we're so interested in can come about. And I think that's going to be one of the themes of my talk, is to try to think about how we can integrate or organize, at least by the end of the talk, maybe you'll get some ideas about how we can try to put all this information together and actually do something to make some predictions. And that's where the modeling comes in, but I'm going to start by talking about some experiments. And I always like to start off by acknowledging the people who are involved. This is work done largely by two graduate students, Judy Bloom and Olena Marchenko. And of course our collaborators, Stormy and Eric, and the people in their lab who've been working most directly with us, Noel and James, and you heard from James already today. And then Michael Blinoff, who's a computer scientist who has developed a lot of the modeling approaches that we use. So it's been really great to have, us all sort of in the same place. Stormy's, of course, as you heard, a great geneticist and molecular genetics, and Eric is an electrophysiologist. My lab is focused primarily on cell biology or cell biophysics, and so you'll see that way of thinking in my talk. So we start off with some experiments. Most of the talk will be on modeling, but I'll show you some experiments that are motivating us, and also that I think are very important. We've taken these iPSC cells, both from Angelman patients and from Dup15q patients, and we've grown them up, differentiated them into neurons, and did some morphological analysis. Somebody asked James about that this morning, so here's the answer to whoever asked that question. So we do 3D imaging after transfecting the cells with the GFP, and very briefly, here are the summary of the results. So a shoal analysis, which looks at dendritic branching. I guess the easiest way to look at this is to look at the three examples on the left. Angelman cells are not very highly branched. The neurotypical cells have a reasonable level of branching at 30 weeks, and the Dup15q cells tend to be quite a bit more highly branched. And the plot that you see over here, let me get this cursor on permanently. The plot that you see over here just compares the distance dependence from the soma in terms of the degree of branching. It's actually kind of interesting. We don't quite understand this, but there's this is over many cells, obviously, and there seems to be some periodicity in the level of branching as a function of distance. So that's kind of interesting too, because it shows that the cells are displaying a fairly internally consistent phenotype. The second kind of analysis that Olena in the lab did was to look at dendritic protrusion density. And in this case, these are aimed at looking at dendritic filopodia and dendritic spines. How many protrusions per unit length are there? And here again is a typical control cell. Here's an Angelman cell that shows quite a few, a smaller number of protrusions, smaller protrusion density. And here's a Dup15q cell at 29 weeks, which shows really a huge number of spines. And this is one example, but here's the statistics showing large numbers of cells analyzed, of dendrites analyzed, or dendritic segments, I should say. So we still have a lot more to do here to see how robust this is, but these are very long-term experiments. As you can see, we had to culture these over many weeks, and anything that goes wrong ruins a year's worth of experiments, so they're difficult. But it looks like we have some very robust effects that are very reminiscent of what people have been seeing all along in mouse models. Okay, so what underlies the morphogenesis of dendrites, the production of branches, the branching morphogenesis, as well as protrusions? Well, it's actin. Actin is the key cytoskeletal element in dendritic protrusions. This is a little composite slide. I'm not going to talk about this any further beyond this slide. It's results that I think Olena presented in this forum previously. But it shows that really that actin and actin dynamics is what controls dendritic protrusions and dendritic morphogenesis. So how do we go from this basic understanding of the cell biology of morphogenesis to trying to understand what goes wrong in either Angelman or Dup15q? Well, there should be a UBE3A connection, right? We know that UBE3A is a key component of the system. So this is a little cartoon that Judy prepared. Thank you, Judy. I think it displays what's going on fairly nicely. And it basically shows you what our hypothesis is. So when a dendritic protrusion, which is what was represented here, finds a bouton on a neighboring axon, it engages some receptors called FB receptors. They release affexin 5, which also gets phosphorylated in the process. So these little blue things are phosphorylation. And then affexin 5 can be degraded in its phosphorylated form by UBE3A. And UBE3A essentially controls the level of affexin 5, so then it gets degraded. And once it's degraded, its usual target, which it acts as a G protein exchange factor, targeting a molecule called Rho-A. That molecule, Rho-A, controls shrinkage of dendrites, okay? So if affexin 5 is no longer there, and I'm sure many of you know about this, then the Rho-A pathway is downregulated, and the RAC1 pathway, which upregulates actin dynamics and actin polymerization, is turned on, and the spine can enlarge. Okay, so that's the basic hypothesis, and so that's sort of where UBE3A might come in dendritic morphogenesis and spine morphogenesis. So this just recapitulates what I've been saying. Under normal circumstances, there's a reasonable balance of Rho-A and RAC1 activity, and you get a reasonable number of protrusions. In the Angelman syndrome situation, you might have a downregulation of UBE3A degradation of affexin, and that would activate Rho-A to a larger extent and cause the loss of protrusions. And in cases where UBE3A is upregulated, Rho-A would be turned off, and the RAC1 system would take over and activate actin dynamics. Okay. So how do we go about trying to really, at least theoretically, see if this is all feasible? Well, we do it using modeling. And I'm going to introduce our modeling approach because I'm hoping to at least maybe inspire a few of you to think about using this approach in your own systems and see if ultimately we can combine forces through the power of modeling. So over the last 20 years, my lab has developed a modeling software system called Virtual Cell, and I should say that it's really not a virtual cell. It's a software tool for modeling cell biology. And this little picture on the side here shows the kinds of different kinds of simulations that can be run. We can solve ordinary differential equations, sort of kinetic systems. We can solve partial differential equations where the kinetic systems are associated with specific geometries. We can do stochastic simulations. We can also do something called network-free simulations, which I'll talk about a little bit more in a minute. We have 5,400 users who have actually used the software, who've built models. It has a graphical interface that is designed for biologists so that you can build your models fairly easily, and the math is automatically generated. The simulators are automatically deployed. It can deal with reactions, diffusion, advection, membrane transport, lateral diffusion on membranes, and even electrophysiology. So it can really integrate a lot of different kinds of mechanisms. And it has, as I mentioned, compartmental, ordinary differential equation solvers, spatial, partial differential equation solvers, and it can also do deterministic and stochastic simulations. But here's the part that I think is really important in terms of sort of integrating the different kinds of ideas that we've heard about today. We've heard about sodium proton transport. We've heard about sodium proton exchange, rather. We've heard about some electrophysiological experiments. We've on KCNQ as being a key molecule. We all know about GABA as being a key molecule in our system. And of course, there's UB3A and the Actin system. How do you sort of keep all that straight? And if you start building the models, you're encapsulating, you're hard coding all this data, in fact, in quantitative form, and you can begin mixing the different features that you know are important in the system. And we have a database underlying Virtual Cell that holds on to all of this modeling information and allows you to copy one piece of a model from one system into your own particular model. We have pathway database support. I'll talk about rule-based modeling. And we can even integrate experimental data into the Virtual Cell software. Okay, so that's Virtual Cell. What are some of the modeling strategies that you can use? Well, you can build a reaction network. So this is a simple reaction network for a system where molecule A interacts with molecule B to form a dimer, AB. B can then interact with C to form ABC. But of course, B can also interact, C can also, A can also interact with BC to form ABC. So there are a lot of different ways that you can get to this final product and several different intermediates that you can use. So this is one way of expressing it, and it's fairly nice because, as I said, it visualizes what's going on. Another way to express the same thing is using reaction rules, where you sort of visualize the molecules that are involved. Here you have an A molecule that can bind to a B molecule through specific sites, a B binding site and an A binding site to form the product. And here there's the B molecule binding to C, again through specific sites. So these two ways of expressing this particular model are completely equivalent. And virtual cell allows you to express your model in either one of these representations. But the second representation is kind of a little bit broader or a little bit more useful when you're dealing with something like UBE3A. And the next slide sort of attempts to try to explain why. So what if, in addition to A binding with B through a particular binding site and B binding with C through a particular binding site, what if B can also dimerize? That is, this site that we had previously reserved for B binding with C, maybe it can also bind to a counterpart on another B molecule to form a dimer. But once that can happen, you can imagine that you can actually form a polymer, because if you think about it, there's a C site still waiting over here and an A site still waiting over here, which may be unbound, and this can continue. So representing this one reaction really represents a polymerization reaction, which can give you an infinite number of, an infinite network. So I've actually generated this network from that simple dimerization reaction, or polymerization reaction, but I've concatenated it so that it doesn't completely fill the universe. How do you deal with a network like that? Something like polymerization or clustering, or in the case of UBE3A, ubiquitinylation, where you can have multiple ubiquitins that are required before degradation. So you can deal with it using a technique called network-free simulations, where you seed the system with a fixed number of molecules, and then you just let the system propagate automatically, so that you stochastically or probabilistically allow for the various bindings based on the rate constants that you know from experiment, and then watch the system evolve. So that's basically how we've approached this problem. And here's the sort of generic ubiquitinylation mechanism that has been encoded in virtual cell using a rule-based model, and then simulating that model with a network-free simulation. So to get to the results, we created this rule-based model. It encompasses the ubiquitin proteasome pathway, but it also encompasses the engagement of the FB receptor with its ligand and the release of affexin and the phosphorylation of affexin, and then following that, of course, the entry of affexin into the ubiquitin pathway for protein degradation. So all of those features have been incorporated into this model, and I believe it's actually the first comprehensive mathematical model of the ubiquitin pathway. There have been sort of lumped models of ubiquitinylation, but nothing as detailed as what Judy Bloom has produced in Connecticut. So here's a screenshot from the virtual cell model, and these are all the different rule-based reactions. These are the reaction rules. I've highlighted one of them showing FB interacting with the FB receptor to produce the liganded receptor, and in so doing, releasing affexin 5. And as you can see, well, I'll show you more in the next overlay, affexin 5 goes from the unphosphorylated form to the phosphorylated form. Maybe you can't see that too well in the back, but this is a typical rule, reaction rule, and is one component of this fairly complex model. So I don't have time to go through all the details, obviously, but just to give you some results from that model. Here's effexin 5 degradation, and we've set up 3 different scenarios. 0 nanomolar UBE3A, what we call a control level of UBE3A, which is 50 nanomolar, and here's Dup15q level of UBE3A. You can see, first of all, that there's very little degradation in essentially we're at steady state. And by the way, since these are stochastic simulations, you have to do several runs and average them, and that's why there are error bars here. So these are.
So this is great. In terms of like how you put that model together, without going into the details, you've built up all the component rules, as I understand it, from published papers and insights from those.
Right.
And
some guesswork.
And some guesswork. Now the way of doing this model, presumably are weighting each of those component rules as building blocks. How sensitive are these results to those weights and those choices you made in the inclusion of the model together? So if you were to weight them a little bit differently, do the charts change?
So in other words, how sensitive are they to the parameters?
Right. You have to sort of guess on.
Yeah, that's exactly the kind of question that you ask, and that's one of the. So first of all, I would say that you don't do modeling in the absence of experiment. You need to use modeling to inspire experiment. So we can do that kind of sensitivity analysis to see which parameters are the most important for the system, and then go back to the lab and measure those parameters specifically in our system. A lot of the reaction rates and concentrations that we get from the literature are not necessarily even in neurons. They may be in other, completely different systems, but they're what's available to get us started. But once we have the model built, then we can start asking questions like that.
I take the assumption that you made is that the only ubiquitin ligase acting on effexin 5 is UB3A.
Right.
Given that there's no data.
Well, again, I mean, it's kind of a generic ubiquitin pathway. So if it wasn't UBE3A, it wouldn't really make much difference in this model.
But I think the thinking is that there are many different ligases that might act on a particular substrate. So.
Sure. Yeah. So yeah. So I mean, there are a lot of assumptions here, but it's a way to get started and a way to start organizing what you know and thinking about it. So right. So getting a little bit to Guy's question, what happens if we change the concentration of UB3A a little bit? Oh, actually, this is looking at degraded effexin. And I think this is important because it's showing that effexin keeps going up because there's a constant level, degraded effexin keeps going up because there's a constant level of synthesis input to the system. So in fact, the degraded effexin is sort of asymptoting to the same level in the presence of both 50 nanomolar and 100 nanomolar UBE3A. Slight variations in UBE3A may aid in spine development. So this goes to the question of sensitivity. Here you see that here's the baseline that you saw before with 0 nanomolar. Just increasing to 10 nanomolar helps quite a bit. And going up to 20 nanomolar almost gets you within standard deviation of the sort of control level. And I think I'll skip that. Just in the interest of time, to get to some of the take-home messages, here's a pathway diagram that you can get off the web for row A. So I told you that affexin, its primary target is as a GEF for row A. Here are all the things that row A can do. One of them is actin nucleation and polymerization. Well, it actually inhibits it, but it has all these other downstream effects as well. And what's really going on in our cells? What's really going on in dendritic spines? Well, the other question is, there's a lot more to actin polymerization than just activation of row A or activation of RAC1. And here's a reaction diagram in virtual cell that encompasses many of the mechanisms in actin polymerization. So this is in virtual cell, so it's in the database. So it is conceivable that we can just take this reaction network, copy it into a UBE3A affexin model, and see what happens to actin. And that, I think, is one of the take-home messages, that there are lots of people contributing to virtual cell, and hopefully, as I said, I'll inspire a few of you today. We have, as I mentioned before, 5,400 users who've run simulations, who've built models and run simulations. We have currently 73,000 stored models on our database. The publicly available models, the models that anybody can look at, are 914. Those are usually models that are based on papers that people have published. And so there's already quite a gold mine, I think, of information, quantitative information on various cell biological pathways and physiological mechanisms. So just to show you how this might be done, down here is our database, and I've done a little search for GTP just to look for other Row or RAC-related mechanisms, and I've gotten several of them. But within a model, I can do a search for Row. I don't know if you can read that in the back because the resolution got lower by going to the slide screen, the projector. But I did a search for Row. I come up with several reactions in various different models that are involved in the biochemistry of Row. I picked one of them, and I can just pop it right into the same model. So this is the search and the pop, and so this is a way that you can use the database to build up more complex models. So to summarize, human neurons from cultured AS and Dup15q stem cells develop respectively deficient and enhanced levels of dendritic branches and protrusions. Regulation of actin dynamics underlies the variation in dendritic morphogenesis. UBE3A may regulate actin dynamics in dendritic spines through the Row-GEF affexin 5. I think it probably almost certainly does, but obviously there's a lot more going on. And we can recapitulate at least the ubiquitin pathway in a virtual cell rule-based model. And while many parameters remain uncertain, simulations show that affexin 5 dynamics and steady state levels are highly sensitive to variations in UBE3A. And finally, and I hope this is my most important take-home message, VCell or virtual cell can serve as a platform for organizing and integrating our knowledge of dendritic spine physiology. So hopefully, I'll be here obviously the next couple of days. If anyone wants an on-the-side demo of virtual cell, I'll be happy to do it. It's online and available for free to anyone. Thank you.
Questions? Questions at all?
Yeah.
Is a virtual cell only for intracellular interactions, or can you use it? Are there tools for modeling network oscillations like the beta activity that Damon was talking about?
Yeah, I mean, it's not restricted to single cells. You can have multicellular interactions. It does have one limitation with respect to electrophysiology. You can model multiple cells. I think that probably should be possible. But within each cell, at any one point in time, the cell has to be isopotential. So you can't model propagation of action potentials down an axon. The action potential happens simultaneously everywhere in the neuron. But you can model action potentials. There's other software. There's neuron software that you can use for that kind of thing.